Good day, dear students. Today our lecture topic cell injury. The plan of our lecture: first question about cell injury, definition, etiology, types of cell injury, stages of acute and chronic cell injury, specific and non-specific symptoms of cell injury, types of cellular death, necrosis, apoptosis, pathogenesis of apoptosis, and other types, the general mechanisms of cell injury, pathogenesis of cellular membrane damage, and the next, ionic imbalance in pathogenesis of cell injury, deficiency of energy in pathogenesis of cell injury, and protective compensatory reaction in pathogenesis of cell injury. And so, cell injury is a damage of structure and function of the cell. Cell injury implies the changes in its structure and function that seriously compromise its viability or the ability to adapt to pathogenic factors. Cell injury have different causes. Here two groups like exogenous and endogenous. Exogenous including mechanical factors, extreme temperature, electric shock, radiation, cyanides, arsenic, mercury, influence, strong acids and alkalis, environmental pollutants, insecticides and pesticides, alcohol and narcotic drugs, and biological like microbes, viruses, prions, fungi, protozoa, parasites, and psychogenic factors, and endogenous, including excess or deficiency of oxygen, hydrogen ion, potassium, calcium, sodium, free radicals, fluctuation of osmotic pressure, influence different metabolites, mediators of damage and mediators of inflammation and allergy, and genetic defects. The cell injury developed after influence different etiologic factors, but here depend on resistance of the cell damage and firstly, a type of cells. Highly specialized cells like neurons and muscular with a high level of intracellular regeneration are resistant or more resistant to damage than other cells. And cells with a low intracellular level of regeneration, for example, blood cells, skin, intestine, epithelium, easily injured. Next point, like a cellular environment influences on the cell resistance for noxious factors, and here microenvironmental induce cell differentiation and proliferation. Fourth, nervous and endocrine regulation. The nervated cells are damaged more easily. The nervous system regulates energetic and plastic processes in the cell. A cell-deprived nervous and endocrine regulation dies by apoptosis, and damaged neurons induce cell injury by producing pathophysiology, which come into cell with exoplasma through exons. 5. Microorganism state. Avitaminosis protein deficiency reduces resistance of a cell to damage. And 6. Phases of cell cycle. The cell is differently sensitive to various etiological factors in different phases of cell cycle, like ionizing radiation damaged cells in phase G1 and G2. And type of cell injury. We know two types, acute and chronic. Depend on time, etiological factors influence it for short time and intensive. And chronic, etiologic factors have a small intensity and influence it for a long time. And stages of acute cell injury, 5 stages. 3 stages, primary specific effect of the etiological factors, non-specific reaction of a cell and necrosis, are reversible, and necrobiosis and necrosis irreversible. Two stages. And stages of chronic cell injury. Three stages. First, emergency stage or alarm reaction. Here, increasing functions of undamaged structures, and after that, activation of cellular genome, activation of protein synthesis. Second stage, CD adaptation, hypertrophy and hyperplasia of intracellular structures, stabilization of RNA, protein, and ATP synthesis, and third, dystrophic changes and destruction of cells. And other types of cell damage, like a direct primary injury, it means damage of cells by etiologic factor, and indirect or secondary injury is the result of primary injury. It's caused by mediators of damage by neural and endocrine disorders. Another type, like a partial and total, reversible and irreversible. Here are some reversible signs. First, plasma membrane alteration, such as blebbing, blunting, or distortion of microvilli, and loosening of intracellular attachments. Second, mitochondrial changes such as swelling and the appearance of phospholipid-rich amorphous densities. Third, dilation of the endoplasmic reticulum with the attachment of ribosomes and dislocation of polysomes. And four, nuclear alteration with clumping of chromatin. And irreversible signs like increased staining. Next, cytoplasm becomes vacuolate and appears more thickening. Discontinuous in plasma and organelles membranes make a dilatation of mitochondria with the appearance of large amorphous densities, disruption of lysosomes, and appearance of intracytoplasmic myelin figures. Lastly, breakdown of DNA and chromatin, and some type of changes of nucleus, like a karyolysis, is a complete dissolution of the chromatin. Pycnosis characterized by nuclear shrinkage and increased basophilia. And karyorrhexis, the pycnosis nucleus undergoes fragmentation. Specific and non-specific symptoms of cell injury. Specific are caused by specific action of etiological factors. For example, cyanides inactivate cytochrome oxidase in mitochondria. Certain bacteria can elaborate phospholipases that degrade cellular membrane phospholipids. Radiation leads to free radical formation, and high temperature lead to protein coagulation. And non-specific symptoms of cell injury are present in every injured cell. Here, for example, high membrane permeability, changed enzyme activity, ionic imbalance, damaged energy supply, intracellular acidosis, and cellular swelling. Types of cellular death: necrosis and apoptosis. And stages of apoptosis. First stage, signaling pathways that initiate apoptosis. Second, program of apoptosis, control and integration processes in which intracellular positive and negative regulatory molecules stimulate or forestall apoptosis and thus determine the outcome. Third, realization of program, a common execution phase including realization of the death program and accomplished largely by the caspase family of proteases. And lastly, removal of dead cells by phagocytosis. Here are some mechanisms of apoptosis. like a receptor dependent, mitochondria dependent, p53 mediated, and perforin enzyme dependent. First, mitochondria dependent pathway. The induction of apoptosis is dependent on a balance between pro and anti-apoptotic proteins of the BCL family. The pro-apoptotic proteins include some sensors that sense DNA and protein damage and trigger apoptosis, and other effectors that insert in the mitochondrial membrane and promote leakage of mitochondrial proteins. Second, receptor-dependent pathway. Here, death receptors bind with ligand, and after that, activation of adaptive proteins, thus domains, and lastly, activation of caspases, and result DNA fragmentation. Next way, perforin enzyme pathway. Here, nature killer cells produce the perforin. Perforin bind with cell membrane and perforate membrane. And after that, bind with granzyme molecules of the target cells and activate the process of apoptosis. Okay, and here some differences between necrosis and apoptosis. Necrosis is a result of causal degradation. Apoptosis in a series of physiologic events. It is not causal but programmed. And main enzymes. Here lysosomal hydrolysis. Here cytosolic caspases. And necrosis accompanied by release of inflammation-inducing intracellular constituents. Apoptosis is not accompanied by a release of inflammation-inducing substances. Necrosis causes damage to adjacent cells by intracellular hydrolysis and indirectly by attracting neutrophils. Apoptosis does not cause inflammation. Cells disappear without trace. A little bit information about the dysregulated apoptosis. Disorders associated with defective apoptosis and increased cell survival. Prolonged survival for reduced turnover of abnormal cells. And like a result, formation of cancer, especially a tumor with p53 mutations or hormone-dependent tumors, such as breast, prostate, or ovarian cancer, or autoimmune disorders, which could arise if autoreactive lymphocytes are not eliminated after encounter with self-antigens. Disorders associated with increased apoptosis and excessive cell death. These diseases are characterized by a marked loss of normal or protective cells. Neurodegenerative diseases manifested by loss of specific sets of neurons, such as spinal muscular atrophy, ischemic injury, as in myocardial infarction and stroke, death of virus-infected cells in many viral infections. And how do you know apoptosis is a single form of programmed cell death? Here, autophagy, life as self-eating, refers to the lysosomal digestion of the cell's own compartments. Cellular stress, such as nutrient deprivation, activate autophagy genes, which initiate the formation of membrane-bound vesicles in which cellular organelles are sequestered. These vesicles fuse with lysosomes in which the organelles are digested, and the products are used to provide nutrients for the cell. Next, like a necroptosis, programmed necrosis, caspase-independent programmed cell death. Necroptosis is triggered by ligation of TNFR1 and viral proteins. Necroptosis is caspase-independent, but dependent on signaling by RIP1 and RIP3 complex. RIP1 RIP3 signaling reduces mitochondrial ATP generation, causes producing of reactive form of oxygen, and permeabilizes lysosomal membranes, thereby causing cellular swelling and membrane damage as occur in necrosis. Release of cellular contents evokes an inflammatory reaction as in necrosis. Next form, like a apoptosis, so-called because it is accompanied by the release of fever, inducing cytokine interleukin-1, and because it bears some biochemical similarities with apoptosis. It is characterized by swelling of cell, loss of plasma membrane integrity, and release of inflammatory mediators. Apoptosis results in the death of some microbes that gain access to the cytosol and promote the release of inflammation-generating interleukin-1. Now we should consider some universal mechanisms of cell injury. As of here, 5 mechanisms. First, damage to cellular membranes and enzyme system, loss of the integrity of cellular membranes. Second, arrangement in the energy supply and utilization. Third, ionic and water imbalance. Fourth, changes in the genome or its abnormal realization. And last, disorders of intracellular regulatory mechanisms. You can see here a normal membrane, a function structure, and function-dependent structure, and some function like a selective permeability, diffusion, membrane palm of penocytosis, cell receptor for cell recognition and communication. And here's a normal structure of membrane. Here, one head and two tails. One head and two tails. For example, here glycerol. It means the head of phospholipids, and saturated and unsaturated fatty acids, hydrophilic heads and hydrophobic tails. Here, for example, choline. And so, pathogenesis of cellular membrane damage, including 4 mechanisms. First, activation of lipid peroxidation. Second, activation of endogenous phospholipids and other hydrolyses. Number 3, osmotic or mechanical injury. And lastly, immune injury or macromolecular absorption or penetration. And first mechanism. Here, 2 main components. First, excessive formation of free radicals, ionizing radiation, ultraviolet radiation, heavy metals, high level of oxygen stress, hypervitaminosis D. And second, decreased activity of cellular antioxidant system, inherited or acquired. You can see here excess of pro-oxidants, and deficiency of antioxidants. Pro-oxidants, vitamin D, NADPH2, NADH2, prostaglandins metabolites, catecholamines metabolites, and metal with different valency transition metals. And so antioxidants, superoxide anion dismutase, superoxide dismutases, catalase glutathione peroxidase, vitamin E, proteins contain SH groups, glutathione, ceruloplasmin transferrin. And so here the mismatches here, more pro-antioxidants and less antioxidants, and results increasing lipid peroxidation. You can see here chain reaction of lipid peroxidation. Initiation is the step in which a fatty acid radical is produced. Here OH group bind with unsaturated lipid, and after that formation lipid radicals. And lipid radicals, and here propagation of chain lipid peroxide radicals, lipid peroxide, lastly formation. As this circle continue and continue and continue. And termination, when a radical reacts with a non-radical, it always produces another radical, which is why the process is called a chain reaction mechanism. The radical reaction stops when two radicals react and produce a non-radical species. This happens only when the concentration of radical species is high enough for there to be a high probability of collision of two radicals of their neutralization by antioxidants. And results here, two tails, and lastly one tails, only one tail, one tail, one tail, one tail. Here defect of structure means defect of function. Information for sample. Here's defect and increasing permeability of membranes. Okay, second one. Activation of membrane phospholipases and other hydrolases. Some causes like ischemia, poisoning of snakes, spider, sting of bees, increased level of intracellular calcium, increased permeability of lysosomal membrane, and results destruction of membrane phospholipids and destruction of membrane cytoskeleton. Third mechanism, fluctuation of osmotic pressure. And in the situation when osmotic pressure of extracellular sector, for example, 400 milliosmoles, and here 300 milliosmoles. And you know results: molecule of water, leaf cells, and moving on to extracellular sector, and results degradation of cell shrinkage. Or a different situation, when osmotic pressure inside cell more than in the extracellular sector. Here swelling of cells, membrane destruction, osmotic hemolysis. Here different consequences of membrane damage. Increased permeability after destruction of phospholipids below. And so leakage of intracellular enzymes across the cell membrane into the serum. For example, here from cardiomyocyte leakage in the extracellular space, some enzymes or specific proteins for contraction proteins, and hepatocytes, here LT, or lactate dehydrogenase 5. Here first. Okay, another signs like a high absorption ability, development of autoimmune process, damage of reception, transport, communication, membrane functions, disorders of ionic transport and energy production, ionic imbalance, decreased in resting membrane potential and action potential, disorders of impulses generation and transfer, and disorders of cellular metabolism, intracellular acidosis. And so here different universal mechanism for damages of cell, total cell. Here the arrangement is the energy supplying and utilization, including three. First, decrease in the rate of efficiency of ATP production. Here including oxygen deficiency or metabolic deficiency, glycose, fatty acids, decreased activity of oxidative enzymes, mitochondrial damage, uncoupling oxidation from ATP synthesis. And B, disorders of ATP transport system, low level of creatine phosphokinase here, KPK, creatine and creatine, low activity of CPK. And lastly, the arrangement of ATP utilization, low activity of ATPases, here low activity of ATPases. And consequences of depletion of intracellular adenosine triphosphate. Here, activation of anaerobic glycolysis, and after that, increased lactic acid, formation of acidosis. Acidosis cause of lysosomal enzyme activation, and lysosomal enzyme activation, and lastly, autolysis or necrosis. Or other effect bind with calcium, increased level of calcium, activation of hydrolysis. Okay, here in other mechanism like ionic imbalance and causes damage to membranes and ATP deficiency, and here decreased membrane potential and developed hyperkalemia, more sodium, increased osmotic pressure in the cell. Osmotic pressure in the cell have effects like a swelling of cell. Okay, here consequences of increased intracellular calcium level. Membrane phospholipids, calcium-dependent proteases activate, activation of phospholipids, membrane damage, activation of proteases, catabolism of structural and membrane proteins, activation of ATPases, acceleration of ATP depletion, activation of endonucleases, fragmentation of genetic material. So many noxious effects. Okay, in other consequences of increased intracellular calcium level, like uncoupling of oxidation from phosphorylation in mitochondria, myofibrils contraction or contracture, and decreased adrenoreactivity. Here are some compensatory reactions in injured cells. First, compensation for the arrangement in the energy supply, like activation of anaerobic ATP resynthesis in the glycolytic process in the cytosol, and aerobic respiration in the intact mitochondria, stimulation of the energy transport from the sites of production to the sites of its utilization, and second, protection of the membranes and the cellular enzymes, including activation of the antioxidant systems, activation of the buffer systems, increased synthesis of the microsomal cytochrome P450 or other enzymes involved in detoxification. And lastly, increased resynthesis of the damaged molecules of enzymes and components of membranes. And the last one, alleviation or removal of water and ionic imbalance, including activation of ionic pumps and repair of membrane. And fourth, repair altered genetic program, including removal of breaks in the DNA strands, excision of the altered sites in the DNA molecule, and restoration of the native DNA fragment in place of the damaged one. Fifth one, compensation for disturbance of the intracellular regulatory processes, including changes in the density of the cell receptors, up or down regulation of the receptor's affinity toward ligands, changes in the activity of adenylate cyclase, guanylate cyclases, phospholipases C, protein kinases, or other factors involved in signal transduction. And lastly, removal of unfolded proteins. Increased in level of heat shock proteins, chaperones. And next, increase in metallothionein synthesis. Metallothionein protect against heavy metal industrial damage and may participate in other fundamental physiological and pathological processes such as antioxidants, antioxidation, proliferation, and cell survival. And next, compensatory mechanism like a decrease in functional activity of cells, regeneration, hypertrophy, and hyperplasia. And here, case for assess how do you understand lecture. And so radiation injury causes cell damage. Which of the listed remedies will promote and which will hinder the development of radiation sickness and why? Okay, here, influence of the radiation and some injuries, acting up some functions of cell. And so if you use sodium and potassium ATPase inhibitors, here for example, sodium and potassium ATPase inhibitors. And after that, how do you know sodium increased in the cell, and after that swelling of cell and necrosis developed. And so next, like a sodium potassium ATPase activator have a protective role against the radiation. Antioxidants, you know, after effects of radiation here produced more free radicals, and antioxidants bind the free radicals. Calcium 2+ channels blockers, you know, consequences. Here, increasing level of calcium. As they use, we use blockers, channel blockers. After that, decreased level of calcium or normalized level of calcium. Okay, yeah, it's a protective role and buffer solution. You know, here decrease the pH and buffer solution normalize the pH level. Okay, and so we have considered main parts of the pathophysiology of cell injury. Thank you for your attention. See you on the next lecture.